I welcome everyone to our COVID-19 vaccine webinar. My name is Jessica Cole, and I am a VEBA account manager. Thank you for taking the time today to join us to learn about the COVID-19 vaccines, such as vaccine efficiency, safety, variants, and why you should get vaccinated. At VEBA, we remain steadfast in our commitment to support you in any way that we can. Before we begin, a few items of housekeeping. To help streamline the presentation, all participant lines are muted. If you have a question, please submit it through the Q&A function. Questions will be answered either directly back to you or for the broader group. They will be held until the end and answered by our panelists. If you wish to not share your name, please click on Send Anonymously when submitting your question. The presentation is being recorded and it'll be available for you to view after the webinar tomorrow on our VEBA COVID-19 landing page. This webinar is intended to be general in nature and will not be addressing individual situations or issues with vaccine availability. For the latest news in your area or medical group, please visit vebaonline.com and click on the COVID-19 vaccination info button. You should see closed captioning subtitles appearing on your screen. If you wish to turn them off, please click on the live transcript button on your screen and select Hide Subtitles. For anyone who has an interpreter, they can call into the webinar to the phone number and webinar ID listed at the bottom of this screen. All right. The last few months have brought us unprecedented and unanticipated change to our daily lives. Our agenda today focuses on the COVID-19 vaccine and trial design, types of vaccines, vaccine challenges, and why you should be vaccinated. We may not have all the answers today, but we will give you our best information so you can make informed decisions. For current COVID-19 vaccination information, please visit vebaonline.com and click on the COVID-19 vaccination info button. Please check this landing page continually as information is constantly changing. I would now like to introduce our presenter for today's webinar, Dr. Susan Little. She is a professor of medicine in the Division of Infectious Diseases and Global Public Health at UCSD. She is the director of the Primary Infection Research Program, director of the UCSD AIDS Clinic Trials Group, Clinical Research Site, and co-director of the UCSD Antiviral Research Center. Most recently, she's taken an active role in the COVID-19 Prevention Network, leading phase 3 efficiency trials for COVID vaccines. Thank you so much. And Dr. Little, go ahead, it is all yours.
Thank you very much. Thanks for the invitation to present. And I will share screen. All right, can you see slides?
Yes, we can.
Small successes. All right, welcome everyone. Thanks so much for inviting me. And I have no disclosures. And as was already mentioned, these are sort of the general topics that I'm going to cover today, focusing mostly on the available vaccines. But as you'll soon hear, we're going to have another one that I think will be joining Moderna and Pfizer within a week. So I'm going to cover a bit on the Johnson & Johnson, also known as Janssen vaccine. So vaccine and trial design. So I think everyone has now seen a lot of the coronavirus structure, which is marked by these spike proteins protruding from the surface. And blown up here is one of the spike proteins. This protein is what interacts with the host receptor and mediates spike receptor binding and internalization of the virus. So not surprisingly, this is the focus, the spike protein of almost all vaccine candidates. This is the target for vaccine candidates for almost all of the vaccines that are in clinical and preclinical development, other than those that involve whole or live virus, which is almost none of them at this stage. So right now, the vaccine pipeline in the US involves 6 vaccines. We're going to focus just on a few of them: Moderna and Pfizer, which I think you all know well, AstraZeneca and Janssen, which are coming up next, and then Novavax and Sanofi. These are all very large studies, 30 to 40,000 individuals who are enrolled on these trials. They involve different platforms. So I mentioned that they all use the spike protein as the target, but they present that target in a different way, which is the different platform, the way in which they present the target to the host, to the person. The start date, we've started a new vaccine trial almost every month since July. And the first data readout refers to the date at which the sponsor can present milestone safety and efficacy data to the FDA and apply for emergency use authorization if their data are adequate. So as you already know, Moderna and Pfizer received their emergency use authorization in November. Janssen is the next company. We'll get to that soon, but they will be reviewed on Friday of this week. AstraZeneca will be a bit later, probably late March, mid-March, and then Novavax will follow soon thereafter. Their study is fully enrolled as of about a week ago. And then Sanofi is a bit further delayed. So a lot of people worry right off the bat. It's just gone too fast. I can't trust a process that has moved this quickly. So normal vaccine production timeline is about 8 to 15 years. And in COVID timelines, also known as Operation Warp Speed, the goal was 12 to 18 months. And Pfizer actually exceeded that and had a vaccine in people in under 12 months. So how was this accomplished? If you look at all of the steps involved in a normal vaccine approval process, there are roughly 6. But even if you add up all of the elements in red, you still don't come up with 12 to 18 months. The only way Pfizer and Moderna were able to achieve their goals in under 12 to 18 months was by overlapping these steps. So they simply were able to do multiple things simultaneously. And what I really want to focus on here are clinical trials and the subsequent approval, manufacturing, and distribution. So what has been done is that clinical trials are divided up into phase 1, phase 2, and phase 3. Phase 1 is safety. Phase 2 is sort of looking for further safety. And in the case of vaccines, immunogenicity. Do the vaccines generate the immune response that we think is necessary to protect people? And then we move into phase 3. Is that immune response effective at preventing COVID? So that's the phase 1, 2, and 3. All of the phase 3 trials that are underway have demonstrated adequate immune responses and thus are going into phase 3 trials. But it's critical that when that phase 3 trial gets to the adequate milestone steps that I just described for Pfizer and Moderna, that they present to the FDA and the FDA says, yes, you've met that milestone, and they approved, not in 6 months, but in a couple of weeks. The manufacturing has to already be done and distribution plans already worked out so that 1 week after approval, we were already vaccinating people. So all of these steps had to be accelerated and overlapped. And that's how this process was done. But no steps were skipped, okay? All of the same safety measures that are done for every vaccine that's been approved since the beginning of this vaccine production process were still completed for all of the COVID vaccines. So for Moderna and Pfizer, this is for both of them, they're roughly the same. This is what the mode of action looks like. So you take the spike protein and you convert it to messenger RNA sequence. So that's just the template that codes for the protein. Naked RNA is very unstable. So they they coat it in this lipid nanoparticle to make it more stable and then package it as a mRNA vaccine. This is essentially the blueprint, and this is what's delivered as part of the vaccine into muscle cells. It arrives there and makes lots and lots of copies of this spike protein using the cell's own machinery. critically important, this doesn't enter the nucleus of the cell. So it's not long-lived in the body. It makes lots and lots of copies, and then these cells die, and that's it. That's part of the reason you get 2 vaccines. You kind of get 2 shots at this, and then the template is gone. The protein fragments are presented at the surface of the cell. These spike protein fragments, where they're recognized as foreign, they stimulate several different kinds of cells, B cells and T cells. And the B cells make antibodies. And the hope is that these antibodies in part neutralize, that is, block spike proteins that are present up here. So these antibodies bind spike proteins when they are present if you ever get infected by coronavirus, and prevent that virus from actually entering a cell and infecting it. So that's the cycle here. So the Pfizer vaccine, this vaccine is called BNT162b2, lovely name. And it encodes, again, the spike protein of the SARS-CoV-2 formulated in this lipid nanoparticle. This trial involved 150 clinical trial sites in 6 countries, including 39 US states. This study enrolled people 12 years of age and older. You probably know it was approved for people aged 16 years of age and older. But keep in mind, there are adolescents aged 12 to 15 who are enrolled in this trial, and data will be forthcoming for this group. It's simply, the FDA did not think there were sufficient data for this group of adolescents at the time they approved the vaccine for people aged 16 years of age and older. So the trial is ongoing. Most of the people enrolled are somewhere here around the 6-month mark. And they will continue out to 2 years, again, looking for safety, efficacy, durability of antibody and immune responses. People were randomized 1-to-1, vaccine to placebo. And after the vaccine was found to be efficacious, they brought people back in, and people in the placebo arm are probably still being vaccinated because it was considered unethical to continue people on the placebo arm. So this is the result slide. Shown here is days after dose 1, vaccine, placebo, and this is the cumulative incidence or number of new cases of COVID. And you can see a huge difference. Cases of COVID plateaued very early in the vaccine arm as compared to cases of COVID, which just steadily increased over time in the placebo arm. And what you can see is this is a vaccine where 2 doses are given at day 0 and day 21. And so if you go up to this blow up here, which is the first 21 days, you can see as early as day 12, the 2 study arms separate. Overall, the vaccine efficacy, this will be abbreviated throughout this talk, VE is vaccine efficacy, was 95% 7 or more days after the 2nd dose of vaccine. So highly effective. This is, a home run in the world of vaccines. And the vaccine efficacy was comparable across ages, race, ethnicities, gender, and underlying conditions. So it was a highly effective vaccine. And that's really the major point of this slide. Moving on to Moderna. The Moderna, again, this vaccine is called mRNA 1273. Also, it encodes the S protein of SARS-CoV-2, also formulated in a slightly different lipid nanoparticle. This was 100 clinical trial sites in the US. They enrolled only adults 18 years of age and older. 2 injections, 28 days apart, randomized 1-to-1 vaccine to placebo, 30,000 individuals. Similarly, they're a little less far along. They started a little after the Pfizer study, so they're somewhere in here. Again, this is going to continue for 2 years. So all of these studies are still underway and will continue. They are also in the process of bringing everyone back in and vaccinating everybody in the placebo arm once the vaccine was shown to be effective. So again, this is the result slide. Shown here is the vaccine arm in blue and the placebo arm in gray. Same result as in the Pfizer study. you can see that cases of COVID over time continued to increase as compared to cases in the vaccine arm. And shown here is beginning 14 days after the second dose, which was on day 28, you can see a huge split between the two arms. The vaccine efficacy here was 94.1%. Again, vaccine efficacy was comparable across age, race, ethnicity, gender, and underlying condition, so highly efficacious. In addition, this vaccine was found to be highly effective in preventing severe COVID. So cases of COVID that led to hospitalization and death. That was an endpoint that was only a trend in the Pfizer study, but highly significant in this study. So I am going to talk about safety, but rather than mention for each individual study that I just presented, I'm going to show a much larger amount of safety data that has followed the surveillance since they've been approved. So to talk about it, first I have to talk about how we measure vaccine safety. So there are multiple ways, but VSafe, for everyone who goes forward to get vaccinated, you'll be introduced, I hope, to VSafe. It's a safety monitoring app that is patient facing. So you'll get it on your cell phone. After vaccination, you'll get a VSafe information sheet that tells you how to register, and the CDC runs this, and you'll get regular queries as to how you're feeling and asked to report daily post-vaccination how you're feeling and to register the types of symptoms that you're feeling. There's also a vaccine adverse event reporting system that medical providers use, and this is to report more serious adverse clinically important side effects that are also reported. And then there's also VaxText, which simply reminds you when to get your second dose. So for both mRNA vaccines, Pfizer and Moderna, for the first month after their use, authorization, VSafe surveillance reported on 1,600,000 vaccine recipients. About half are Pfizer and half are Moderna. So I'm going to show you the data here. This is just for Pfizer, and I want you to only look at the blue lines to begin with. This is dose 1, blue lines. Local and systemic or whole body side effects were equal between Pfizer and Moderna. So the same relative amount of side effects for both vaccine. One doesn't have more or less than the other. So because they're about equal, I'm showing you Pfizer. So let's look down, and yes, the side effects are a bit more than you would get with your typical influenza or pneumonia vaccine. So injection site pain, where you get the injection, about 70% of people had injection site pain, but it's mild. The vast majority of people have mild pain. Same with fatigue, headache, muscle pain, fever, joint pain, chills, nausea, swelling, all the way down. These are percentages. 29% of people had fatigue. So the vast majority of all of these bars are mild, and then there are some people that had moderate and very few that had severe. When you look at the side effects, the people who had severe disease, more people who had the, I would say, severe, intense side effects are younger people. And here, younger is defined as under 60 to 65. So you're young if you're under 60 to 65. And younger people are more likely to have more intense side effects. So one difference between Pfizer and Moderna was that the side effects for the second dose were greater after the second dose for those who got the Pfizer vaccine. So that's why I'm showing you the orange bars. So if you got the Pfizer vaccine, when you got the second dose, it actually went up. So fatigue now went up to 50%, headache 42%, and you can see the orange bars. So the intensity and the frequency of the side effects actually increased a bit with the second dose for the Pfizer vaccine. And that was not so much for the Moderna vaccine. Independent of these side effects, both of these vaccines have been associated with, unfortunately, some life-threatening allergic reactions in the medical field known as anaphylaxis. Early on, the rates were a bit higher, but now after there've been, 1 million plus doses given, these seem to be settling out in about this range. For Pfizer, the rate is about 5 cases of anaphylaxis per million doses given. And for Moderna, it's about 3 cases per million doses given. And I'll give you a bit more data on sort of what that means later. But by comparison, for an influenza vaccine, the rate is about 1.5 cases of anaphylaxis per million. And for the the pneumococcal vaccine, which people get at age 65 and older, the rate is about 2.5 cases per million. If you look at all vaccines, not these two, all other vaccines as a whole, the rate is about 1 case of anaphylaxis per million. So these are higher, there's no doubt about it. So just to touch on some common questions that I come up every time I present this, so I'll just knock them off right off the bat. What is the allowable time window between vaccine doses? Because there's been a lot of noise in the news. The CDC has come forward mostly to fill the void because there's a lack of data, saying that 6 weeks, 6 weeks after your first dose. So if you're Pfizer and the dose is 21 days apart, it doesn't matter which vaccine you got. 6 weeks after your first dose is probably as far out as you want to go. We're not talking about AstraZeneca today. There are data to suggest that pushing things out a bit further may be more advisable, but we're not talking about AstraZeneca today, and that may be where some of the noise comes from. There are simply no data for Pfizer or Moderna to push that window further. There are justifications to push it further, but there are not data to push it further. How long are you protected after receiving a vaccine for the prevention of COVID-19? Again, I showed you on the timeline, those studies, people are only out about 6 months or 5 months. So, you know, the data are pushing as fast as they can. And what we know are right now, the best data I have are at least 90 days. But, you know, every couple of weeks, there's a new paper. So tomorrow there may be a paper saying that it's a bit longer. What we know is that at least 90 days, people seem to have neutralizing antibody, that is antibodies that bind and prevent infection, that are effective. The titers, the amount of those neutralizing antibodies do seem to decline a bit even over the 90 days, but that's not unexpected. The whole goal of most vaccines is that you rely not on circulating levels of protective antibodies, but you rely on a memory function of your immune response, so that when you get exposed to the pathogen again, these memory cells wake up and generate antibodies again. So far, everything looks good. How long are you protected after having recovered from natural COVID infection? There are lots of different estimates, but in my opinion, the best papers currently suggest about 8 months. And then should I receive a vaccine if I've already had COVID-19 infection? Yes, mostly because we don't yet know whether natural COVID infection gives you the same level of protection that a vaccine offers. We do know that people can get COVID twice. And so at present, the recommendation is yes, get vaccinated. But in this time of vaccine shortages, we do know that people who have already had COVID have probably got protection for some period of time. And so that, 8 months. So the recommendation is consider delaying vaccine for, I don't know, 90 days is often the time limit given, because the likelihood of getting a second COVID infection within the first 90 days is extremely low, because you've got natural protection. So again, yes, get vaccinated, but again, vaccines are short, so you're probably not at the front shouldn't consider yourself to be at the front of the line in terms of your risk. Can children receive vaccines? Unfortunately, no, because we don't have them. We've got the Pfizer vaccine that goes down to age 16. Pfizer does have data on in studies for adolescents 12 to 15. Those are being studied, and they will have those data soon. And based on those data, they will develop a clinical trial for younger children. Moderna is actually launching, they may have already launched it this month or next, a study for children. that's going to go all the way down to 6 months of age through 15 years of age. And that study will be divided into 2 parts. The first part will be dose ranging to find the best dose, and age ranging, dose per each different age range. And it will look at, everyone will get vaccinated, but it will look at what is the best dose that elicits, that generates the best immune response, and with the best safety profile. Once they find that for each age range, they will then move to the second phase, which will be a randomized blinded study, meaning some kids will get placebo. I believe it is a 3-to-1. 3 people will get vaccine to 1 who gets placebo. And that will be an efficacy study, and that will take place at Rady Children's, among other sites in the country. So that will be the Moderna vaccine, and that, again, I believe is starting now, if it hasn't already. Can a vaccinated person develop asymptomatic infection? Yes. We don't have a lot of data here. Again, Moderna has a very small amount of data. The likelihood of getting asymptomatic infection is probably greatest early on after the first vaccine, or within the first, and for Pfizer as well, after the first vaccine, or early on in the first 1 to 2 weeks following the second vaccine, before you're fully protected. And all we know right now, both companies, all the vaccine companies, are looking at this question. We do know that there is a reduction in the number of cases of asymptomatic infection in the vaccine arm compared to the placebo arm, but small numbers. We also know, in terms of the next question, can a vaccinated person transmit COVID? In these cases where, I mean, first of all, yes, anybody with that has COVID can probably transmit. We do know from looking at the Pfizer data, vaccinated people appear to have lower viral loads. So it does appear that the risk of transmitting would probably be lower. But the whole question of transmission requires a different kind of study that has not been done yet. So no one can answer the question of actual transmission, because that involves looking at contacts who have acquired COVID, and those studies have not been done. But I would say there are some viral load data that are compelling to suggest that the risk would be lower. So now we're going to move on to Johnson & Johnson and Janssen Pharmaceutical. They paired together. I'm going to call it Janssen, but you may also see it referred to as Johnson & Johnson. So this study is a completely different kind of vaccine. This is an adenoviral vector. Ad26, adenoviral serotype 26, is a replication incompetent adenovirus. It's a human adenovirus which causes the common cold. So what they've done is they've taken this common cold virus and they've modified it so that it is replication incompetent. It can't replicate. And they've inserted into it the coronavirus spike protein. So there's lots and lots of data with this platform. So they've used this platform for a lot of other vaccine studies. For instance, Janssen has used this adenovirus vector to license an Ebola vaccine in Europe using this Ad26 vector. So they have a lot of experience with this. But specifically looking at coronavirus, in earlier studies, side effects were mild to moderate. One very unique feature about this vaccine study is that it's the only one that has a single injection. Single injection, 2 years of follow-up, again, 40,000 people. This is a truly international study. 10 countries are participating. They submitted their interim analysis January 29th, and looking at across all 10 countries, 28 days after the single vaccine efficacy was 85% in preventing severe COVID disease. This is the only study also that has what we say powered or analyzed their data specifically for the prevention of severe disease. It's great if you can prevent mild to moderate disease, but what we really want to do is prevent death and serious life-threatening disease. There were no COVID-related hospitalizations or death after day 28 of the vaccine. Protection was generally consistent across race and age groups. There were a third of people over age 60 across all variants and all regions. And I'll get to talking a bit more about this. In the US, which was 44% of the enrolled population, vaccine efficacy was 72% in preventing moderate and severe disease, meaning it didn't work as well in preventing moderate disease. So it was 85% for severe, 72% for moderate and severe, 66% globally for moderate and severe disease. But one thing I'll ask you to put in your memory banks, because we're going to come back to this in a bit, vaccine efficacy 89% against severe disease caused by the South African variant. The South African variant is the one that everyone is worried about now and has just recently entered the US. And we'll talk about this a bit more. So knowing that we have a vaccine that works well against the South African variant is a big deal. Vaccine was well tolerated, no notable safety signals, lower side effects, reactogenicity in older folks. This is the same as all the vaccines. Side effects are fewer in older individuals compared to younger individuals. The most frequent adverse events, also known as side effects, were headaches, chills, fatigue, and muscle aches, all at relatively low numbers. So this is their timeline. They submitted the data I just showed you were January 29th. They submitted their package on February 4th. They submitted for emergency use to the WHO on February 19th, and this Friday, they will be reviewed by the FDA for VEBA, as we refer to it, rather than this long name. And we anticipate emergency use authorization as early as next week. So vaccine challenges, and really here I'm going to move kind of quickly because this is kind of beyond the scope, but there's so much in the news lately about these variants of concern. So you don't need to learn these names. These are the UK variant, the South African variant, and the Brazilian variant. And really the things we need to know about them are the UK variant is expected to be the predominant variant in the United States within the month of March. So it's expected. And it is about 50% more infectious and does appear to have an increased risk of death compared to non-UK variant viruses. That's why if you're following things, you've started to see the CDC talk about double masking because this is a more infectious variant. The South African variant has also it's become the predominant strain in South Africa. It does not have any evidence of increased or altered disease severity, but it does appear to reduce the effectiveness of some vaccines. And the South African variant seems to be similar in terms of the mutation profile to the Brazilian variant. So this is the UK variant and where it has now been detected in the US, and again, expected to become predominant within 1 month. This is the South African variant where it's been detected, and this is the Brazilian variant. So, you know, why am I so worried about the South African variant? It's only been detected a few places. Well, because we don't measure very well. The US, this is the sequencing. In other words, to detect variants, you have to have a good surveillance program. And that means you need to sequence isolates of the coronavirus as they occur. The United States is down here at number 43, which means we sequence 0.3% of our cases. Britain, the reason we know about the UK variant is because Britain sequences about 7.4% of their isolates. Most experts think you need to sequence somewhere around 10% of your isolates to have an adequate idea of the circulating variants. So we may have more or fewer South African variants. We simply don't know. We don't sequence very many of our isolates. Now, there has been a big push. We've recognized this problem, and the current administration has just released $200 million, I think, to ramp up sequencing. And I think there's a bill on the floor of the House to push that up to something closer to $1.7 billion. So this issue is being addressed, but just to recognize at present, when you see these numbers being discussed, we simply don't know what's out there at present. So how do the current Pfizer and Moderna vaccines work against these new variants? Because they were tested at a time that the variants didn't exist in the US. And so the best we can do at present without redoing the studies is to look at people who've been vaccinated and take blood from them and test how well those sera, the blood from those individuals who's been vaccinated, can neutralize this UK and South African variant. And so they did that. They pulled isolates from serum from 12 people who were vaccinated with Moderna and 10 people who were vaccinated with Pfizer. And what you can see is white is good, red is bad. People who had been vaccinated with Moderna and Pfizer over here. And what this shows is that there was no loss of neutralizing antibody activity against the UK variant. So everyone except this one person, because 2.5 was the level, almost everybody was able to neutralize this variant if you'd been vaccinated by one of these two vaccines. However, every sample had decreased activity against the South African variant. And so the expectation is, and the rough fold drop was, I don't know, call it 10-fold drop for both of them. And the expectation is that the Brazilian variant will behave quite similarly. So no one knows how this translates into vaccine efficacy, but it is expected that there will be a drop to some extent, but I don't know if that's from 95% down to 93 or from 95 down to 85. No one knows that. But that's why we're watching this South African variant so closely and the Brazilian variant. So I'm not going to run through answers to all of these, just to rather say that there are still a lot of unanswered questions. We need to know what protects people. What are the immunologic signatures of protection from SARS-CoV-2? And ideally, these would serve as endpoints for future vaccine trials. We'd like to know if there are going to be less common side effects, adverse events that, we'd like to monitor over longer term to see if any of these longer term side effects emerge. I'm not going to talk about enhanced disease, so I won't talk about that. Does vaccination reduce asymptomatic infection? We think it may, but don't have a lot of information. And then transmission, we need a totally different kind of study to look at this. But clearly this brings up the need that even post-vaccination, we're going to continue to need to mask and social distance to some level until we've got a lot of people in the community vaccinated, because we still don't know the answer to this question. What about vaccine safety and efficacy in other populations? Children, pregnant, immunocompromised. We don't have answers in those populations. What's the durability? I already explained we don't have great answers on that. And will circulating variants compromise vaccine efficacy? So I'm going to end with a few words on vaccine rollout. We know, unfortunately, that certain populations have been much harder hit with COVID, and these sort, unfortunately, by race and ethnicity. So if you look at American Indian cases, hospitalizations, and death, you can see the increase. 4 times the rate of hospitalization, 2.6 times the rate of death. Same for Black, African American, and Hispanic individuals, much higher rates of hospitalization and death. And why is this? Well, I'm going to skip. In addition to that, before I get to why is this, in addition, what are the risk for hospitalization if you have any of these conditions? Asthma, 1.5 times higher, hypertension, 3 times higher, and so on, all the way across. Obesity, if your body mass index is greater than 30, 3 times, but severe obesity, 4.5 times. If you have 2 conditions, 4.5, 3 or more conditions, 5 times. So again, there's a sort of a graded scale of increasing risk of severe disease if you have any of these conditions or multiple of them. So what are the factors that contribute to all of this? Social and structural factors impact COVID-19 risk. People of color are overrepresented in essential service industries with increased exposures. Low-wage jobs often do not provide health insurance, paid sick leave, childcare, etc. Residential segregation. People of color are more likely to live in residentially segregated settings with high housing density, poor access to healthy foods, more multi-generational households with limited space, and increased levels of chronic stress resulting from structural racism, increased incidence of violence, and everyday aggressions and trauma. So how do we overcome vaccine hesitancy? We're going to start with this. This is the most recent survey I could find, a National Pew survey in November of 2020, showing that 60% of Americans said that they would get the vaccine probably or definitely, with another 39% who said no, with some of them who might be convinced. So why? Why is this number so low? Well, there's a lack of trust in government and healthcare systems. We have a terrible past, a history of systemic racism and unethical experimentation in certain populations. And this mistrust must be addressed with respect and honesty. We must engage vaccine hesitant subgroups with culturally sensitive and tailored risk communications and messaging. We should involve faith and influential community leaders, increase knowledge levels, reduce perceived risks, and enable informed decision-making, and ultimately try to increase confidence in vaccine safety and effectiveness. So second, a lot of people are, again, as I said earlier, concerned that the process has just moved too fast. The vast majority of the data that was presented to the FDA for approval included data out to 60 days after the second dose of vaccine. And that's because the vast majority of adverse events or side effects occur within the first 42 to 60 days following immunization or vaccination. And that's why they limited the data. They wanted to move things along fast, but also include the information that was likely to include the majority of side effects. If there's a concern that the review process wasn't transparent, in fact, it is extremely transparent. The Pfizer review and the Moderna review are entirely available on C-SPAN. You can click on these links, you can search them, you can see all of the data, you can watch the debate, public comment, the deliberation, you can see everything that was presented. Each of these links has about 8 hours of deliberation and debate, and you can see the entire thing. Religious concerns. There are no human fetal or stem cell tissues used in the mRNA vaccine development at any point in its design or development. In contrast, the Janssen vaccine may contain human fetal or stem cell tissues. So for people who have this concern, important to know this information. Where are we now? So we're vaccinating now, phase 1B. We're vaccinating tier 1 individuals 65 and older, and just announced today, educators. We're supposed to be vaccinating March 1. We hope we have vaccine. So to be determined, I hope. But across the state, vaccinations will be beginning. So who can receive the COVID vaccine? And it's important to recognize I didn't say indications for vaccination because there really aren't any. It's who can receive. Healthy adults, yes, people with immunocompromised conditions, cancer, HIV, transplant recipients, etc. There's really no contraindications here as long as you are relatively healthy. Autoimmune conditions, lupus, rheumatoid arthritis, etc. Pregnant and lactating women, people who have a history of Guillain-Barré syndrome, if you have it, you know it. People with dermal fillers, this can cause some inflammation and swelling at the site of the fillers, but this is not typically problematic. It can be treated with some steroids. A history of anaphylaxis not related to vaccines or injectable therapies or components of the mRNA vaccine. So you can be allergic to pets, venoms, food, bees, oral medications. Those are not contraindications. People with current or prior SARS-CoV-2. So if you've had COVID, again, I mentioned this earlier, you don't want to get vaccinated while you're actively ill. You have to wait until you've met criteria to discontinue isolation. And while vaccine supplies are limited, maybe temporarily delay vaccination. Who should not receive vaccination? If you have anaphylaxis or an immediate allergic reaction within 4 hours of any severity to a previous mRNA vaccine, Moderna, Pfizer, or any of its components, you should not get vaccinated. If you have an immediate allergic reaction of any severity to polysorbate or polyethylene glycol injected, not oral. Polyethylene glycol is in everything. So injected, you should not receive the vaccine. That's it. It's a pretty limited list. Also, a note on co-administration with other vaccines. Typically, we want 14 days. If you're going to get a flu vaccine, go ahead and get it. Try and separate it by 14 days with your COVID vaccine. However, a shorter period is okay if the benefit of the other vaccine outweighs the potential unknown risk of co-administration. What that means is if you just had a measles exposure, you need that other vaccine. That's important. So, again, there are times when the benefits of the other vaccine are critical and you need to get it, a hepatitis exposure, et cetera, epidemic. So talk to your provider. I mentioned this briefly, California Notify. It's a great feature that will help you know if you've ever been exposed to anybody who has who has COVID. And it's a completely confidential program. Your privacy is protected, your identity is not known, your location is not tracked. If you have an iPhone, you simply turn on the notification already in your settings. If you have an Android phone, you have to download the app. But it's very cool, you can read about it. Future directions, last slide. Vaccine development and widespread implementation is really our best hope for pandemic control. Community engagement efforts are critical to ensure that our communities are well informed, that there is community support for research, that we have meaningful relationships, reciprocal partnerships, and increased trust. And we still, unfortunately, need to continue to adhere to what we call non-pharmacological interventions, social distancing, mask wearing, hand washing, et cetera. And I know a lot of people get really frustrated, like, why am I getting vaccinated if we need to keep doing this? Things are going to continue to get better incrementally, but we still, unfortunately, have some unanswered questions, and we can't just get vaccinated and rip our masks off. Sorry. So I'm going to end there, and Jessica, I am done.
Perfect. Thank you so much, Dr. Little, for that presentation. You provided us with so much information. At this time, I'd like to go ahead and address some questions you may have. So if you do have a question, please submit it through the Q&A function. Let me go ahead here. Perfect. And one question that we do have for you, Dr. Little, is have there been any fatalities from getting the vaccine?
I am not aware of any fatalities. There was a fatality actually in LA, of all things, last week, but it was not associated. It was temporally linked, in close timing. Somebody was at the vaccine center, received, an older woman, I think, received the vaccine. She died, but they do not believe that was related to vaccine at all. It was not an anaphylaxis reaction. So I have not seen the final, but at the time, soon thereafter, they did not feel that it was a vaccine reaction or vaccine related death, which I can only assume mean they think she had some unrelated cause of death, but I haven't seen the details on that follow-up.
Thank you. Can you get the first vaccine shot from Moderna and then the second shot from Pfizer, or vice versa?
Not advised. What the CDC recommends is in exceptional circumstances that can be done. And I think what they mean by that is if you have gotten one vaccine and it is impossible to get the second vaccine within 6 weeks, there is no supply, and you do not have the ability to get vaccinated, you could under those circumstances. But it is strongly preferred to stay within your lane.
Perfect, thank you. And what happens if your second shot goes past that recommended time due to availability?
That's a good question. I think at this stage, no one is suggesting that you start over. So I think the answer would have to be consult with, your local provider and discuss it. Because I think the answer is it depends on how far over. So yeah, if you're 12 weeks over, that may be much different than if you're 6 weeks and 3 days.
Gotcha, perfect, thank you so much. And how long do the side effects from the COVID-19 vaccine typically last?
Should have said that. The vast majority of side effects occur the day after the vaccine. So don't think you're home free if you feel great the day of vaccine. And most of them last 2 to sometimes 3 days, but most people have 24 hours of feeling eh. So the day of vaccine, they're okay. The day after vaccine, they don't feel great. And the following day, they're on their road to recovery.
Perfect. Thank you. And personally, which vaccine will you personally get or have gotten, and why did you choose that vaccine?
My answer is always you pick the vaccine that you have access to. So, and be grateful. but but I got Moderna. Yeah, so I honestly believe they're both incredibly equivalent. I confess, in the studies, both Moderna and Pfizer showed an increase in the frequency of side effects at the second dose. In the much larger VSafe data that I showed you, Moderna did not show an increase in the frequency of side effects at the second dose. So I don't know, maybe you see how much you want to have side effects, but I think there's a reasonable likelihood that people are going to have side effects, especially younger. And again, COVID side effects, they're not that bad. I had a sore arm. I had a 1-day headache with the second vaccine. And I should also mention, which I left out, it is not advised to take preventative Tylenol, Motrin, Advil, etc., prior to your vaccine to try and prevent these side effects, because there has been some data to suggest that that can limit the immune response. So if you need to take those things to cope with the side effects afterwards for medically indicated reasons, totally appropriate. But don't take Motrin, Tylenol, etc. in advance of your vaccine.
Gotcha. Perfect. Thank you so much. Now, I know you went over briefly the types of vaccines and what they are, but are all COVID-19 vaccines the same regardless of the company that produces them?
No. So Moderna and Pfizer are messenger RNA vaccines. They're the same type of vaccine produced by 2 different companies, but they're very similar. Janssen, also Johnson & Johnson, is an adenoviral vector. So it's a totally different kind of vaccine. And it is very similar to the to the AstraZeneca vaccine that you've heard about in the news, because AstraZeneca has been approved in the UK, in all of Europe, in the WHO. AstraZeneca will likely be approved in the US in late March, is my guess. It'll be the 4th one. So AstraZeneca and Janssen are very similar. They're adenoviral vectors. So that's that modified common cold virus. And then the last one, which I didn't even get to, I'll come back in a couple of months, Novavax, which is a protein subunit. So this is they all use spike protein. So they're all going to generate the same type of immune response. It's just how they present it. It's the way they elicit the immune response. But they're all going to generate what we hope is anti-spike antibodies.
Perfect. Thank you. Now, a person who's been vaccinated, can they be contagious? And should they still isolate for 2 weeks? And why do people think that this myth, if it is a myth, can be true?
So if you've been vaccinated, you are not fully protected until you are probably 1 to 2 weeks after your second vaccine. So until you are 1 to 2 weeks after your second vaccine, you should assume that you are likely, you can get COVID. So when I talked about people getting asymptomatic infection, I mean, the people who get COVID, let's just say most of the infections occurred in the between the first and second vaccine, or within the first week of the second vaccine, because you're not fully protected. So you should keep doing all the same things you've always done: socially isolate, mask, hand wash, etc. And I don't want to say when you're fully vaccinated, stop doing those things, but you will feel a lighter sense of relief. But yeah, you're not fully protected until 1 to 2 weeks after your second vaccine. So yes, you can get infected.
Perfect, thank you. And what was in the placebo that was used in the trials?
Saline, salt water.
Salt water, perfect.
That's it.
And basically is, let's see here. Also, I want to remind everyone too that this is being recorded and will be available on our landing page tomorrow. You would be able to view it as well. And let me see if we have any questions. Oh, we do. Can someone with a pacemaker get the vaccine?
Absolutely.
Perfect. Thank you. I believe that's all the questions that we currently have right now. I'll go ahead and bring this screen back up. Perfect. So thank you again, Dr. Little, for your presentation today, along with all the information that you have provided us about the COVID-19 vaccine. For further information, please visit our VEBAonline.com and click on the COVID-19 vaccination info button. If you have any additional questions, please contact the State of California COVID-19 Information Hotline at 833-422-4255. They are available to take your call Monday through Friday from 8 a.m. to 8 p.m. and Saturday through Sunday from 8 a.m. till 5 p.m. You can even email them at COVID call center at cdph.ca.gov. And I'll go ahead and leave the screen up if you want to go ahead and write down that information. This will conclude our webinar for today. Again, thank you everyone for attending and joining our webinar. We hope that you take away a lot of information on the COVID-19 vaccine. Thank you again, Dr. Little, for joining us and presenting all that information. Please, everyone, stay safe and have a wonderful rest of your week. Thank you so much.